Board, so thank you for reminding me. Okay, here we are, chapter 9, and our topic is meiosis. Our topic is meiosis. So here's what we have been talking about so far. Here's what we've been talking about so far. We've been talking about the cell cycle, and we basically, we saw the cell cycle, and you've seen me draw this diagram before, where there's the interphase, and then there's the mitotic phase. And here we have G1, S, and G2. And this is all, if I'm typing this out, this is all interphase. And then, of course, this right here is mitosis. Okay. Well, I cannot emphasize this enough. Okay. I cannot emphasize this enough. But the purpose of mitosis is for cell replacement. It is for wound healing. This is how you get more cells. And it's also for to support physical growth. So that at the end of the cell cycle, is two identical daughter cells. Two identical daughter cells. Now, what cells undergo meiosis? Or sorry, what cells undergo mitosis? Let me make it clear here that we're talking about the cell cycle. So what cells undergo mitosis? And the answer is all cells at least once in their lifetime. Okay, all cells, at least once in their lifetime, they will undergo mitosis. Some cells will only undergo mitosis. So that's the only way they reproduce. Most cells will use mitosis as their method of reproduction. But there's a special type of cell that doesn't use mitosis exclusively. Okay. There's a special type of cell which uses an alternate to mitosis. And that is called meiosis. So there's an alternate to mitosis called meiosis. The end result are 4 daughter cells that are genetically distinct one from another. So this is one huge difference between this mitosis versus meiosis. In mitosis, the daughter cells, there's only 2 of them. Here we've got 4. In mitosis, the daughter cells are identical, and I should put that they are genetically identical. They're clones. Okay, they're clones if you want to think of them that way. They are genetically identical. Here, not only do you have 4 cells, but each cell, their DNA is different. Their DNA is different. So you don't get clones. Everybody is a little bit different from the other. So who undergoes meiosis? And the answer is what we call gametes. And that's another word for sex cells, or in other words, sperm and egg, or what we call ova. So we often say that mitosis is for body cells, whereas meiosis is for sex cells or gametes. So when we say who undergoes meiosis or mitosis, you can say body cells, also known as somatic cells. So like your skin cells, your fat cells, your muscle cells. But when it comes to specifically your sperm and your egg cells, they undergo a different type of cell division, something called meiosis. So where does meiosis fit in the cell cycle? Meiosis fits right here at G2. Okay. So sex cells only, instead of undergoing mitosis, they kind of take a little side road and then they come back and they do meiosis. They skip mitosis and they go meiosis. And then at the end of meiosis, they join here. So kind of like they bypass mitosis and then they come back here. I'm going to talk about the stages of meiosis, and I'll be honest, they're going to sound a whole lot like mitosis, but I'm going to do my best to point out the differences between mitosis and meiosis. Those of you that are following along in the slideshow, I would tell you that we are starting here on slide number 9. Okay, slide number 9. If you're following in your book, we're on page 155. So if you're following along in the stages, check out slide number 9 or page 155 in your textbook. So a few things before we start describing the stages of meiosis. Meiosis is part of what we call sexual reproduction. And in sexual reproduction, you require 2 parents to produce 1 offspring. The parents are each, and if I draw a little diagram here, here's parent number 1, and here's parent number 2. Their DNA is unique. Parent number 1's DNA is also unique. And then when they form a new offspring, the offspring's DNA is unique. So a cool thing about sexual reproduction is that everybody's DNA in this diagram is different. Mother Nature, not only does she want you to pass along your DNA, that's the primary purpose of reproduction, but she also wants the DNA to recombine, or she always wants the DNA to do something new. So Mother Nature, and I'll write that out, wants to pass on unique, recombined DNA. She wants the DNA to always kind of be slightly mixed up a little bit, because it turns out that recombining or mixing up DNA, that's good for the environment. It's good. Always keeping the DNA exactly the same is not always good. Now, in the case of mitosis, we do that because when you replace a cell, you want to replace it with exactly what you had before. But when you're creating brand new individuals, new organisms, you want that DNA to mix up a little bit, because it turns out that having slightly different DNA in every generation helps to protect the generation in some sense. It's part of your immune system to slightly change the DNA from one individual to the next. So this is good. So in sexual reproduction, you have two parents and everybody's DNA is different, both of the parents, and then when they recombine to form someone new, well, then their DNA is also different. Let's compare that to, and we're going to do a lot of mitosis comparison. Mitosis is an example of asexual reproduction. Mitosis is asexual reproduction. And what I mean by that is you have one parent, two offspring, and the DNA remains unchanged. So in meiosis, or excuse me, in mitosis, if I draw a diagram, you literally have one cell that divides itself into two daughter cells, and the DNA is the same for everybody. Nobody's DNA looks any different. Well, normally, when we're talking about multicellular organisms, we don't create new humans through mitosis. We create new humans this way. We don't create new humans this way, but this is what we do for that list I keep giving you. So I hope you know you're going to see it on a quiz. But for cell replacement, wound healing, and for physical growth. Meiosis is how we produce the sperm and the egg, which will eventually fertilize in order to make a new human. So meiosis is not making a human, it's producing the sperm and egg, which eventually will undergo fertilization to make the new human.
Miss Weir,
Yes.
can you like put the paper a little bit more down? No, you're okay.
There we go. I just
Thank
noticed
you.
it on my screen. Thank you for reminding me, Erica. So type a number 1 in your screen when I can move on to the next page. Oh, and I forgot to mention to you guys at the very beginning, I'm expecting a phone call from my mom's doctor. They were supposed to call me before we started biology class, but they didn't. So in the event that the doctor calls me during biology class, I absolutely have to answer my cell phone. So I apologize in advance if my mom's doctor calls during lecture. So go ahead and type a number 1 into the chat window whenever you have copied this page, and I can move on to the next one. Oh, good. Okay. I didn't want to go too fast. Thank you, Erica, for letting me know that I had kind of made it hard for you to see. So, okay, now we are going to talk a little bit more. I got a couple more definitions for you. A couple more definitions. In humans, our chromosome number is 46 or 23 pairs. Our chromosome number is 46 or 23 pairs. Okay. So you have 46 chromosomes or you have 23 pairs of chromosomes. When we look at those chromosomes, okay, the reason that we say you have, 46 pairs is because chromosomes come in pairs. And if you've ever seen a picture of chromosomes, what's called a karyotype, a picture of chromosomes, they're always put out in your order. So you have like chromosomes number 1, number 2, number 3, number 4, all the way down to the 23rd pair. Well, chromosome pairs are numbered so that the largest is pair number 1, and then the smallest pair is pair number 22, and then the 23rd pair is called your sex chromosomes. 1 through 22 are referred to as your autosomes. Okay, 1 through 22 are your autosomes. The 23rd pair are your sex chromosomes. Well, the reason they come in pairs is because one was donated by your mom and the other one was donated by your dad. So this is donated by parent number 1, and this is donated by parent number 2. So literally, your DNA is a combination of your parents. So for every single pair, one came from your mom, one came from your dad. Every single pair. When we look at your chromosomes, so let's say if we examine closer, pair number 1. Okay, this is pair number 1. So this is number 1 from your mom, this is number 1 from your dad. We refer to them as a homologous pair. Sometimes we call them homologs. So a homologous pair or homologs. It just means the same chromosome, but from different parents. The same chromosome number, I should say. It's the same chromosome number, but from different parents. So like, for example, in my diagram up here, these would be homologs, but they're chromosome number 2. These are homologs, but number 3, homologs number 4, all the way down the line. So homologs just means the same chromosome number donated by your parents. Now, when they appear like this, just a single strand, we say that it is an unduplicated chromosome, or we can say it's a single chromatid. During the S phase of the cell cycle, remember that you undergo DNA replication. And so you make a copy of that one chromosome, and now you have the original and you have its copy. It looks like a puffy letter X. Looks almost like a puffy letter X. So now you have a duplicated chromosome. And a duplicated chromosome is made up of one chromatid and its sister chromatid. And the place where the two chromatids touch, let's see if I have enough room, centromere. There we go. The place where the two chromosomes touch is the centromere. Right, so a little explanation of what we're going to see in our drawings. So in our drawings, when we see something like this is a homologous pair or homologs that are unduplicated. If we take those same homologs and we draw them like puffy letter X's, those can be homologs that are duplicated. So a lot of vocabulary words here, these are words that we're going to use when we're drawing out our stages of mitosis or meiosis. Okay, a lot of details on this page. Let me know when I can go on to the next page. So type a 1 for me when I can go on to the next page.
Can you go down just a little bit?
Oh, I did it again, didn't I? How's that, Brandon?
Oh, the other way. Sorry.
Oh, the other way? This way.
Okay, yes.
Hey, Miss Weir.
Hi, how's it going?
Good. Sorry for logging in late. I was working. I apologize.
Oh, well, we're glad you're here. We're glad you're
Thank you,
here.
Miss.
All right, so moving on to the next kind of part. This is where, if you have your book handy, this is where it's a good place to start on page 155 or slide number 10. Ronaldo, I'm going to send through the chat window one more time since you just arrived, and I'm glad you let me know because now I can send the file again. I don't think if you log in after I send the file, can you still see it, Ronaldo, or is your chat window blank?
What was that again, Miss?
I sent a file earlier. Can you still see it, or is your chat window blank?
No, ma'am. I'm not able to see it. It's blank.
Okay. Let me send it one more time so that you can have the same thing that we're looking at.
Thank you.
It should be coming up here. It's a big file, but it's coming up in your chat window.
Okay.
All right, so meiosis is divided into two divisions. Meiosis is divided into two divisions. There's meiosis 1, and then there's a little brief period called interkinesis before we go on to meiosis 2. So interkinesis, it depends on if we're talking about males versus females, but there's a stage between the first and the second division called interkinesis. We'll talk about those differences too. But we'll go through first, what are the stages of meiosis 1? Well, the stages of meiosis 1 are prophase 1, then we have metaphase 1, and you probably get the idea, anaphase 1, and telophase 1. And I promise you, this is going to sound a whole lot like mitosis, but there are some major differences. So during prophase 1, chromosomes are long, loose, and stringy, so they exist as chromatin. Chromatin condenses into chromosomes. But two special events that happen in prophase 1 also occur, and I'm going to write them in red. Synapsis, which means all homologs pair. So in other words, every chromosome number 1, or the chromosome number 1 finds its partner. Chromosome number 2 finds its partner. So even though the chromosomes are condensing, this is a unique event that you only see happen during prophase 1. It doesn't happen during mitosis. It happens only in meiosis, and it happens only in prophase 1. Okay, so this always happens. The second event sometimes happens. And the second event is what's called crossing over. And crossing over is when non-sister homologs, they exchange segments of DNA. So this sometimes happens, but not always. And I'll show you a diagram to kind of explain what we mean. But synapsis always happens, crossing over sometimes happens. Why these two events are so significant is they are the source, or at least one source, of what we call genetic diversity. This is one place that we mix up the DNA. It's not the only source, but it's one source of genetic diversity. So it's a source of genetic diversity. So let's look at your slideshow, and I'm going to look at slide number 12. To kind of explain what is crossing over. So here is the diagram from your book, if you're following on your book, or slide number 12. And this is an explanation of what is crossing over. So we have two chromosomes, and instead of standing up, they have them laying down on their sides. I kind of wish they would have them standing up. I like to see chromosomes standing up, but they have two chromosomes, and they are homologs, meaning they're the same chromosome number. So these could be both chromosome number 7s. But the reason that they're different colors is they're trying to distinguish that one came from one parent and the other one came from the other parent. So these are two partners or two homologs that are lying next to each other. When partners or homologs lie next to each other, that's called synapsis. Okay, so synapsis is when chromosomes pair up with their partners. This always happens. The next part sometimes happens. The next part is crossing over. And so the middle part of this diagram shows one leg of one of the chromosomes kind of laying over the leg of the other chromosome. And in the last picture, you can see that these two legs switched places. So now each chromosome is a mixture of both parents. This chromosome is mostly purple, but it has a little green leg. And this one is mostly green, but a little purple leg. What it's trying to indicate is now we have exchanged information between non-sister chromatids. Even though these are duplicated chromosomes, let me go back to the top picture. The purple is in a duplicated state. The green is in a duplicated state. So you have two purple sister chromatids and two green sister chromatids. But the green ones are not sisters to the purple ones. They're homologs, but they're not sisters. To be a sister chromatid, you have to be on the same chromosome. What has happened in the last part of the picture is that we can see the exchange when it occurred was between non-sister chromatids, so that we have chromosomes that are not 100% from one parent now. Each chromosome is partially from parent number 1 and partially from parent number 2. This last result is what we mean by crossing over leads to genetic diversity. We've mixed up the DNA, or you can say we've recombined the DNA. We've mixed it up. Recombination is a fancy way of saying that you mix up the DNA. So those events happen only in prophase 1. They're significant events. You might see questions on a quiz over what are the special events of prophase number 1. The answer is synapsis and crossing over. Metaphase 1, in every metaphase, chromosomes align in the middle of the cell. But what's a little bit different on this one is that you have homologs that are lining up in the middle of the cell. So instead of having like a single file line in the middle of the cell, you have two rows of chromosomes forming the metaphase plate. There's also what we call the law of independent assortment happening here. And I'm going to go to the diagram on slide number 14 to kind of explain independent assortment. So I'm going to look at slide number 14 to explain independent assortment. So here is a diagram of the 4 possible ways that our chromosomes could line up. So in our previous picture here, I'm going to go back. In our previous picture, we're looking at a pair of homologous chromosomes that have undergone crossing over. So here's our chromosomes that have undergone crossing over. We're also going to follow the path of 2 more sets of homologous chromosomes. These have not undergone crossing over, but these ones have. If you look real close, you can see that they are a mixture of purple and green. Well, this is one way that we can line up all 6 chromosomes during metaphase. this circle over here represents another possible way we can line up those 6 chromosomes during metaphase. And if you look real closely, the way these are lined up is not exactly the same way those are lined up. It's similar, but it's not exactly the same. The difference between the two is in the third pair. If you look closely, the third pair are reversed. There's another possible way that we could line up these chromosomes, and that's what this circle is trying to indicate. And then there's a fourth possible way that we could line up those chromosomes, and that's what this circle is trying to indicate. So what the law of independent assortment says is the way that one pair of chromosomes line up does not affect the way the other pairs of chromosomes line up. So let me type that on the screen here. Oh, that's not what I wanted to do. I wanted to put some text in there. Nope, not add a signature. Oh, here we go. This is what I want to do. Okay, that's what I want to do. So here's what the law of independent assortment says is the way one pair of homologous chromosomes aligns at metaphase plate does not influence how another pair of homologs align at the metaphase plate. So the way that one pair aligns doesn't influence, there we go, now you can see it all, does not influence how another pair aligns. So each one of these is a possible way that the 4 chromosomes or the 3 chromosome pairs could line up. So the end result is that there are multiple ways that they could align at the metaphase plate. These are 4 possibilities, only one will occur. Only one possible way will occur, but the result is it will greatly impact the way things are inherited later on. Red. So I wanted to type these little notes on the screen for you so that they would kind of align with what we are looking at. So there's only, even though these are 4 possible ways, not all 4 possible ways are going to occur at once. Only one of these 4 possible ways is going to occur. And whichever one does occur is going to have an impact on how things separate and eventually how things are inherited. So let me know when you are ready for me to move back to my sheet of paper by typing a 1 into the chat window for me. That's real helpful. Just realized I misspelled that. Influence. There we go. All right, good. Good, I'm getting some responses. Okay, so I'm going to switch back to my paper now. So what happens after metaphase 1 is, of course, anaphase 1. And what's important during anaphase 1 is that, and I'm kind of running out of room here, is that we're going to separate homologs from each other. This is important because this is what's going to reduce our chromosome number. And we're going to reduce it by 1/2. You'll also hear this described as going from diploid to haploid. The word diploid means what's your full chromosome number? The word diploid means what is your full chromosome number? And for humans, that's 46. Haploid means literally your half chromosome numbers. And in humans, that's 23. Anaphase is then followed by telophase. Oops. Which is kind of like prophase in reverse. All the things that we did in prophase, we're going to undo in a sense. And the end result, and of course I don't want to forget cytokinesis, also happens. The end result is you have 2 daughter cells that are genetically distinct because of crossing over. And they have half as much DNA as when they started. I probably should have written that in red. But the end result is you have 2 daughter cells that are genetically distinct and they are haploid. We're going to take a gander at slide number 18, and I'll show you what this process looks like. If you're following in your book, this is pages 156 through 157. All right, so let's look now at the slide number, here we go. Looking at slide number 18 from your book. So when we look at slide number 18, here is prophase 1, here is metaphase 1, here's anaphase 1 and telophase 1. This, by the way, is a plant cell. We can tell because of the new cell wall that's being formed. Here's the cartoon diagram kind of showing what's happening. So in prophase 1, and by the way, your book takes excellent notes for you in this section. In prophase 1, we're showing that all the homologs are pairing, so that's synapsis. Notice that they're also crossing over. See how their little legs kind of overlap? So this pair is crossing over, and looks like that pair is crossing over. The other events of prophase are also happening, like the centrosomes, which is these little things right here. They're going to start to form those little threads, or what we call spindle fibers, that will eventually attach to the chromosomes. Then in metaphase 1, those 4 chromosomes have lined up so that they're with their partner, and they've formed the metaphase plate. This right here shows one of the possible ways that they could have lined up. In this diagram, we have the big purple one on the bottom, and in this part, we have the big purple one on the top. It was quite possible that the 2 purple ones would have been on the left or on the bottom, or the 2 purple ones would have been on the top. It's by mere chance that they lined up this way, and it's by mere chance that crossing over happened. So this is why we say this is kind of independent assortment. One of the possible ways that this pair lined up had no impact on the possible ways that the second pair lined up. But what is going to eventually happen in anaphase is that we're going to separate members of a pair. So one big chromosome will go in this direction, one little chromosome will go in that direction. If the 2 purple ones had been on the same side, then the 2 purple ones would have moved in the same direction. If the 2 green ones were on the same side, then the 2 green ones would have moved in the same direction. But these lined up so that the purple ones were not on the same side. So the end result is during anaphase, you have one mostly purple one mostly green one moving towards one pole, and basically the same happening towards the other pole. During telophase, the spindle fibers will detach. That's one of the things that we said is like prophase in reverse. Cytokinesis begins. Now, I don't know if you noticed this, and I was going to write to the publisher, but the cartoon diagram right here, this is of an animal cell. The way that I know that is because this little indentation right here, that is a cleavage furrow. You don't see cleavage furrows on plant cells, but the diagram or the picture of an actual cell that's doing this is a plant cell. If I was a student, I might not have caught that. So I think I'm going to write to the publisher and say, hey, you might let them know, for students anyway, that this is an animal cell and that's a plant cell. But that's just me being a teacher in that diagram. So the end result of meiosis 1 is that you have 2 daughter cells. Notice that the DNA combination is not the same. Okay, the chromosomes are not exactly the same mixture of colors. And also, each cell has half as much DNA as the one that we started with. Though the one that we started with here has 4 total chromosomes, this one only has 2, and this one only has 2. So this is why we say that the daughter cells are haploid. In other words, they have half as much DNA as what we started with. I'm going to go on to the next slide, okay, the next half of the diagram, which is meiosis 2. Meiosis 2 is most like mitosis. It's most like mitosis. The only difference is that instead of one cell going through the process, you have 2 cells going through the process. So let me write this down so that, because I'll get carried away in my talking. So here we go, meiosis 2. It is more like mitosis, except you have 2 haploid genetically distinct cells doing this at the same time. Oh, that's crooked. Sorry about that. When the paper's crooked, y'all let me know. If I was a student at home trying to copy my teacher's notes and the paper was crooked, that would drive me nuts. The stages of meiosis 2 are prophase 2, metaphase 2, anaphase 2, and telophase 2. Since I'm running low on time, I'm going to refer you to slide number, what slide is this? I think this is 18. Or page 157 of text for the events. So I'm going to go back to your slideshow here. I'm looking at slide 18, or slide 19, sorry. I said it was 18. This is slide number 19 now. Slide number 19. So this is the second half of meiosis. And notice that now we have two cells in every diagram. And they're doing the same thing at the same time. The only difference is that their DNA is half as much as when they started, and their DNA is not genetically identical. So here's prophase 2, and notice that again, the spindle fibers are being formed by the centrosomes. Those spindle fibers are eventually going to attach to the chromosomes. Every time we say metaphase, whether it's metaphase in mitosis or metaphase in meiosis, things always line up in the middle of metaphase. So we have something lining up in the middle here. But what's different about metaphase 1 is, again, there are two cells undergoing metaphase 1, and there's only one row of chromosomes down the middle. Every time we talk about an anaphase, we're splitting something. We're not reducing the chromosome number again because these are sister chromatids. All we're doing is we're separating sister chromatids from each other. So during anaphase 2, sister chromatids are separated. That's an important thing to realize. We're not separating homologs, we're simply separating sister chromatids. Eventually, this will become its own set of DNA, this will become its own set of DNA, and the same is here. This will become its own set, this will become its own set. So again, in our diagram, we can see that this is an animal cell because of that cleavage furrow that's forming. If it was a plant cell in our cartoon diagram, we wouldn't see a cleavage furrow, but we can see the formation of a new cell wall happening in our plant diagram. When telophase is complete, now we have 4 cells. Each one of them is genetically distinct, and each one of them has half as much DNA as when we started way back in prophase 1. I'm going to show you a short video, and I decided to share it with you here in lecture. This video is about 6 minutes long, so I have just enough time to share this video. Before I share the video, let me finish up my thoughts here. 4 daughter cells. Each is genetically distinct, and we have half as much DNA versus the parent. That's what we mean by haploid. So I've got about a 6-minute video to share with you. The reason I wanted to share this video is I don't see all of you in lecture. I'm sorry, I don't see all of you in lab. And this video is posted in lab. This video is posted in lab, and I do ask you to watch it in lab, but I thought I'll share it with you in lecture. That way I can make sure we all watch the video. So it's about 6 minutes in length. Where's my video with my window? Here we go. Hello? This is Christina. Oh, can I put you on hold for 1 minute? Thanks. Okay, hold on, guys. I know that, okay, hold on. I know that you can't hear. Let me fix it.
Hi, it's Mr. Anderson, and in this video I'm going to go through the phases of meiosis. Meiosis is a lot like mitosis. It starts with interphase, but remember, the point of mitosis is to make 2 identical cells. And in meiosis, what we're trying to do are make 4 genetically different cells because they're destined to be gametes. They're destined to be sperm and egg. And that's the whole point of sex. We want the next generation to be different than the generation before. And so when you look at a diagram of meiosis, it's a little bit daunting. And we're not going to go through all of it right now. We'll go through this diagram at the end, kind of as a way to review it. But I want to step through each of those phases of meiosis. A quick mnemonic, PMAT times 2 is going to remind you the different phases that we have to go through. So it's prophase, metaphase, anaphase, telophase, and then we go through that again on the second division. Before we get to it, we should talk about the major structures that you're going to see as we go through meiosis. The first one, of course, are the chromosomes. And so you're going to see 2 of each chromosome at the beginning. And so we get a chromosome 1 from our dad and a chromosome 1 from our mom. We call these homologous chromosomes. Remember, each of these chromosomes has hundreds of genes on it. And so you get 2 copies of all those genes, 1 from dad, 1 from mom. This would be chromosome number 2 because it's shorter. And in my model, I'm just going to use 2 pair of chromosomes. Remember, in a real human cell, we're going to have 23 pair of chromosomes. But it's almost too difficult to follow what's going on if we had that many chromosomes. You just have to multiply it times 13. Now, lots of times you'll see chromosomes not look like that, but like this. And so what's happened here is that we duplicated that chromosome from dad. And so these 2, what are called sister chromatids, are exact copies of the DNA. And so you know when you see a chromosome that looks like this, they've already gone through that duplication. Another important structure is the centrosome. The centrosome is going to organize the spindle, which is essentially dividing the nucleus and also dividing the cell. And in animals, it's made up of 2 things. We have the centrioles on the middle, and then these microtubules that go around the outside. In plant cells, they're going to lack these centrioles in the middle, and the nuclear membrane is actually organizing a lot of this division. But let's begin at the beginning of interphase. So this is just as the cell has been formed, and you can see right here in the nuclei that we have those 2 pair of chromosomes. chromosome 1 and chromosome 2. And if we look at the centrosomes, we just have one centrosome. And so that's not usually what a cell looks like. This is what a cell usually looks like. And so what's gone on here, you can see that in interphase, we've duplicated those centrosomes. So we have 2 of those. And the DNA is all loose. It's not tightly wound up into these chromosomes that we characteristically see. As we go through interphase, what eventually happens is then we can see those chromosomes again. Now, do you remember what it means when they look like this X? It means that during the S phase of interphase, we've copied the DNA. So we have 2 complete sets of DNA. And this looks identical to mitosis, but it's just about to change. And so what happens next is prophase 1. During prophase 1, the chromosomes undergo what's called synapsis. And so what's happening is chromosome 1 from dad and 1 from mom are coming together, and they're wrapping around each other really, really tightly. And what's really going on is that they're swapping parts of their chromosomes. In other words, these are pretty much identical except for the changes in the genes. And so they undergo what's called crossing over. So segments of chromosome from mom are switching with chromosome from dad. Same thing over here, and same thing with chromosome 2. Now, what's that giving us? It's giving us variation. If this didn't occur, if synapsis didn't occur, the chromosomes that you get from your mom and your dad, you would give to your children, either as a chromosome from your mom or a chromosome from your dad. And what we're doing in crossing over is we're combining the 2 chromosomes that we got from our 2 parents and making a brand new chromosome that we want to deliver to our child. That's the importance of this. If we keep watching what happens next, we then go into metaphase 1. What's happening in metaphase, you can see that they're all lining up or meeting in the middle of the cell at what's called the metaphase plate. Now, they could have organized themselves like this with the blue one on the left and the red one on the right, but they could have easily organized themselves like this. So they could have been in a different position. So this would be a totally different orientation of the chromosomes. It also could organize like this for chromosome 2, or it could organize like this. And so what do we have? We have 4 different ways that these chromosomes could orient themselves independently at metaphase 1. What is that giving us? Well, it's giving us variation. And so there are 2 raised to the number of pairs, possibilities of how they could arrange at independent orientation of metaphase 1. So how many are there? We would say 4. What if I had 3 here? Then there would be 8 ways that they could arrange themselves. Still doesn't seem like much variation. But remember, in humans, we have 23 chromosomes. And so there are over 8 million ways that all those chromosomes could independently orient themselves during metaphase 1. And so that's where we're getting, again, that and crossing over is giving us a huge amount of variation in meiosis. And remember that one sperm has to find that one egg. And so it's really over 64 trillion possibilities of an offspring just based on independent orientation itself. Let's keep watching then. So another thing that happens in metaphase 1 is that spindle is going to attach. So you can see that the centrosomes move to either side of the cell, and the spindle attaches to the centromere of each of those homologous chromosomes. During anaphase, it's pulling them apart. So we can see that those homologous chromosomes now are going to either cell. And then during telophase, what's happening is we're reforming a new nuclei at each side. We've divided the nuclei, which is meiosis, but now we have to divide the rest of the cell, and that's called cytokinesis. And so we're done with meiosis 1. We've gone through prophase, where we saw that synapsis. We went through metaphase, where we had that independent orientation. And now we've divided the nuclei into each of those cells, but we're not done yet. So what's going to happen next is we're going to go through prophase 2. Now, during prophase 2, there's no more crossing over, but what's going to happen is those 2 chromosomes are going to line up. Again, they're going to meet right in the middle, and the spindle is going to attach to each of those centromeres. Now, if we look what happens, each of those chromosomes are being pulled to another side during anaphase 2. And then finally, during telophase and cytokinesis, telophase 2, we've created these 4 cells that we wanted in the beginning. And so if we look at where did we begin, again, way back in the beginning, we had 4 chromosomes, or 2 pair of chromosomes. Now we have 4 cells, and each of those only have 2 chromosomes, a 1 and a 2. And so what would happen next in the circle of life? These would fertilize a sperm, or excuse me, if these were sperm, they would fertilize an egg, and then we'd start over again. We'd have a brand new organism that's going to be created through mitosis. Now, this is how we make the sperm. Each of the sperm are going to be made like this. Again, there'd be way more chromosomes in us because we have 23. It's a little different with the eggs because there's so much important material in the cell that only one of these nuclei will actually be used and the other ones won't be used genetically in that cell. And that allows us to keep all the important parts of the cell, like the mitochondria, endoplasmic reticulum, in that one cell. And so now let's kind of review and go over that confusing diagram at the beginning. So what are we looking at here? This would be interphase at the beginning. You can see we just have one copy of all those chromosomes. This would be at the end, so you can see that we've duplicated the DNA. Next, what do we have? This is prophase 1. To remember what important thing is occurring there. We've got crossing over, and then we've got independent orientation during metaphase 1. They then are pulled apart, and then we have 2 cells. And now in this second meiosis, what are we doing? Is we're just lining up those chromosomes, and then they're splitting up into each of the sides. And so what do we get at the end? Each of those cells, if we were to go back to here, each of those cells, look at this one, and that one, and that one, and that one, are totally different than that original cell. They also have half of the DNA that original cell did. And so that's meiosis, and I hope that was helpful.
Hello, I'm back. I kept you guys a little bit over time, but I apologize for that. But I really wanted to share that video with you. I'm going to stop the recording.